Okay, thanks very much for the invitation to speak. I'm actually not going to be talking about any human variants specifically today, but what I do is develop and then use high throughput microfluidic assays to study the role of residues throughout enzymes and throughout entire proteins. And I hope to make the argument today, and hopefully that'll stimulate some discussion, that this type of approach is valuable for looking at human variants within coding regions of proteins and enzymes. And it's a direction I'd like to take my lab in, so it'll be a real fun discussion. But basically, to start off, why do we want to study residues throughout enzymes? And so to date, the reality is that most traditional biochemical studies have only focused on small numbers of active site residues or active site adjacent mutations. And the active site is, of course, where catalysis occurs, substrates bind there, and they undergo the chemical transformations there. But more distal residues are also intimately linked to enzyme function, and it's the role of these non-active site residues that are currently the hardest to predict. So just as sort of a very trivial example, a knowledge of the role of distal residues would help us understand and predict allostery, which Monod once called the second secret of life. So a lot of human enzymes are allosterically controlled, and allostery requires the action of distal residues by definition. Sort of away from the interests of you guys, but maybe interesting more generally, is that lots of distal enzyme mutations also turn up in directed evolution experiments when people are trying to engineer better enzymes, but we lack a good ability to predict which sites and which substitutions are going to have the best functional effects. So these experiments just require a lot of luck and in reality, a lot of labor. And sort of highlighting our lack of knowledge is this plot on the right here, where we're comparing the catalytic proficiencies of natural enzymes to de novo and enzymes subjected to directed evolution. And you can see right away that the natural enzymes are like 15 to 20 orders of magnitude better than our best designs. And that's in part because we lack a better predictive understanding of what changes to make and where to make them in enzymes. And then so a more interest to probably you guys from a human health perspective, distal variants turn up all the time in human patient sample sequencing. So just as a small example here are all the C bioportal variants of unknown significance in the human phosphatase, protein phosphatase 1, and they're shown on the structure of the enzyme here. And we can see right away, they're not just at the active site, they're all over the entire enzyme. And currently, we can't predict whether these distal mutations actually have functional effects and if they're actually linked to disease. And of course, if they are linked to disease, future treatments will depend on a functional understanding of their effects. And so there are two challenges to why we don't know a lot about the function of distal residues. And one of these challenges is a conceptual challenge, which I've already kind of alluded to, and the other is a technological challenge. So the conceptual challenge is that the textbook idea or model of enzyme function is that enzymes have these small active sites where the reactions happen, and the rest of the enzyme, which people colloquially refer to as the scaffold, is more or less inert. But in reality, active sites are buried in these networks of hydrophobic, electrostatic, and hydrogen bond interactions. And just by looking at structures, we can't tell which of these interactions are functionally important or what function, if they are, what they actually help achieve. So an imperfect analogy here, but what we want to do is to take natural enzymes apart piece by piece and ideally see what each does, and then ultimately use these data to generate better predictive models for the roles of residues in real enzymes. And so this brings us to the technological challenge, and that's that right now, traditional biochemical methods are just too laborious, too time-consuming, and too expensive to make and study more than a handful of mutations at a time. So for example, for an average enzyme of 500 residues, if you wanted to mutate every position and measure its effect, it would take about 3 years and cost about $500,000 or pounds. And most of that is labor. And so realizing the need for this for new technologies, in my postdoc back at Stanford with Dan Hirschlag and Polly Fordyce, we developed a new technology called HTMEC, where we can do this both in reduced time and at vastly reduced cost. And this is what I'm going to tell you about today. So very, very briefly, we use custom microfluidic chips shown here, and in reality, they're about the size of a postage stamp. And in this region here, if you can see my mouse, the red region, there's 1,500 microchambers in that region. And in each of these microchambers, we can express, purify, and then quantitatively measure a different enzyme variant. So here's how it works. we first just print a plasmid library of mutant enzymes on a glass slide. So these gray circles represent spots of plasmid DNA. We can then align our microfluidic device on top so that each plasmid spot is in its own microchamber. And so each chamber is kind of dumbbell shaped. One chamber holds the plasmid, and the other chamber is what we're going to use as the reaction chamber. And in light gray here are a series of valves on the chip that provide control over fluid flow over the device, and they prevent cross-contamination between chambers. So the next question is, how do we actually make enzyme from the DNA? Well, we just flow in a cell-free expression system, a transcription translation mixture. We use that to solubilize the DNA. Then we close these valves and just put the entire chip on a hot plate and let the enzyme express for an hour. And each of our mutants is C-terminally EGFP tagged, and we use that tag for purification and quantification. So the purification goes like this. Because of the control over fluid flow, we can specifically pattern an anti-GFP antibody under these circular valves here, which we call the button valves. And this means that after expression, we can actually pull down our GFP-labeled enzymes. Then we can kind of close the valves. The analogy is like stepping on a garden hose. We push the valves down onto the glass slide, effectively removing the enzyme from solution. Then we wash out all of the unbound material we no longer want. So this is the purification step. And then we get these nice spots of EGFP, which allow us to quantify actual enzyme concentrations in each chamber. To measure the functional effect of the enzyme mutations, we simply close the valves, flow in a fluorogenic substrate, isolate the chambers, and then start the reactions by opening the button valves and just watch fluorescence build up over time. And by doing that, we can fit rates and quantify enzyme kinetics. So here's how we analyze the data, and we're going to go through the analysis pipeline very briefly just for one chamber containing one mutant. So in the first step up here, we quantify the effective enzyme concentration from the intensity of the EGFP spot. So in this case, we have about 4 nanomolar of enzyme. Then just like in a normal biochemistry assay, we measure product fluorescence standard curves. We just flow in a series of different product concentrations to each chamber, quantify the fluorescence for each one, and then fit a standard curve. And then we're ready to do assays. So in our first assay, we pick one substrate concentration, in this case, 10 micromolar. We introduce the substrate over the chip with the valve closed. Then we open the valves and just measure the rate of reaction, and we fit initial rates. And this is exactly how people do it in traditional biochemistry. We're just doing it in higher throughput. And so because the enzyme's immobilized, once the reaction's done, we can remove it from solution again, flow out the product, and then iterate over a series of new substrate concentrations and then basically measure initial rates for each one, and then use these data to fit Michaelis-Menten curves to get the gold standard parameters of traditional biochemistry, Kcat, Km, and Kcat over Km. And so that's the technological basis. And to demonstrate its sort of utility, we first chose to apply it to PAF-A, which is a member of the alkaline phosphatase superfamily. So these enzymes hydrolyze phosphate monoester substrates, shown at the top right here, and they have the advantages of not being particularly interesting from a biological level, but they're actually some of nature's most proficient catalysts. So PAF-A has a 10 to the 27-fold rate enhancement. It's also a really stable enzyme, so we don't really have to worry about a lot of equilibrium unfolding effects. And we also have a wealth of functional and structural data from the Hirschlag lab and others that are going to help us. Those data are very much focused on the active site, and then we can use that as sort of a leverage point when we look away from the active site and ask how the distal mutations connect to and work in concert with the active site. And so just to belabor the point, it's actually such a good catalyst with this 20 to 27-fold rate enhancement that the reaction it catalyzes, phosphomonoester hydrolysis, would actually take longer than the lifetime of the universe to occur spontaneously. So it's just a really powerful enzyme. And so shown here is a schematic of the transition state of the reaction. And so the key features to note are that there's this threonine nucleophile, T79 in green, and this sits at the N-terminus of this green helix here, which we call the nucleophile helix. And in the transition state, this is attacking or forming a bond with the phosphorus, and then an alcohol is leaving at the same time. Now, this transition state is stabilized by the 2 positively charged zinc ions, as well as these 3 residues here, N100, R164, and K162. K162, and they contact what are called the phosphoryl oxygens. And K162 and R164 in yellow sit on the N-terminus of this yellow helix here. So at this point, a question that sometimes comes up, and it was actually came up in a grant review I recently got back, is that are we just doing a lot of mutagenesis here? And how does this actually help us achieve a predictive understanding of enzyme function? So I'm going to tell you one short story from my postdoc on how we used HTMEC to further understand how PAFA functions at the molecular or physical level. And so the key feature here that allows us to go beyond just mutagenesis is that we have this extensive chemical toolbox of different substrates and different inhibitors that probe different aspects of function. So the real power here is the ability to perform many different assays all on the same chip in a highly quantitative manner. And the important thing to note is that each of these substrates or inhibitors each reports on a different aspect of the enzyme's function, and it's the quantitative comparisons of the effects that can tell us how the enzyme works. So I've asserted that residues throughout entire enzymes are functionally important, but if you just take an enzyme and look at the structure, we don't know which positions are important, where the important positions are, or how many important positions there are. So the first thing we want to do is perform unbiased mutagenesis over the entire enzyme. So to do this, we mutated each position in PAFA to valine and glycine. PAFA is about a 500 residue enzyme, so that gives us about 1,000 total mutations. And as expected, the most deleterious effects on catalysis are in and around the active site, shown here as blue spheres. But when we look beyond the active site, many positions throughout the entire enzyme have an effect when mutated. And the important thing to note is that without these data, we wouldn't have been able to predict anything about what the shape of this distribution looks like, where the big effects were outside of the active site, that is, or what their magnitudes were. But now we get to the point where we have this map, but even having it, we don't really know what these different mutations are doing to compromise catalysis. So that brings us to our next question, in that what sort of specific molecular function is altered? Can we obtain a more sort of granular or specific understanding of how each of these mutations compromises function? And like I said in the last slide, to do this, we're going to use that toolkit of different substrates and inhibitors. So the question we wanted to ask first was which non-active site or which scaffold residues are functionally coupled to the two active site residues I told you about, K162 and R164. So as I said, these residues sit at the N-terminus of this yellow helix, and they donate hydrogen bonds to this phosphoryl oxygen, which we call oxygen O2 or oxygen 2. And so this structural model makes a prediction that we can test. And the prediction is that if the functional role of K162 and R164 is solely to interact with this oxygen, these residues should provide no catalytic benefit for a diester substrate, which has a methyl group here. So you have a methyl group here, and those hydrogen bonds aren't as strong. And consistent with this, mutations to K162 and R164 alone or together decrease the monoester rate relative to wild type, which I'm showing here in yellow, but leave the diester rate unchanged. This is shown here in blue, and you can see there's no change from wild type. And that basically, because these two residues can't help the diester reaction because they can't donate hydrogen bonds to that methyl group, when we remove them, there's no effect from this substrate. And that's consistent with the model I just told you about. Now, conversely, if we mutate this residue N100, which contacts the other oxygen, O1, common to both substrates, we see an equal drop on the rate of both reactions. Again, because this residue is doing something to catalyze both of these substrates. Now, remember, the overall goal of the project here is to figure out what the distal effects are doing. How do these distal sites that have functional effects connect to the active site? So we can actually leverage this specificity effect between monoester and diester and use it as a mechanistic tool to ask which of them are functionally linked to either K162 or R164. And we call this differential effect functional component 1, which is just the differential effect on catalysis for the two substrates. So to illustrate the point I'm making here, let's consider one second shell residue, Y112. So Y112 contacts R164 directly, and when we mutate Y112 to either glycine or valine, it has about a 10-fold effect on Kcat over Km for the monoester substrate. Now, if Y112 is functionally coupled to R164, we expect a drop in monoester hydrolysis relative to wild type, and that's what I'm showing on the right side here. But we expect no change in diester hydrolysis because we already know R164 has no effect on diester hydrolysis. Conversely, if Y112 is functionally coupled to a different residue, for example, N100 or the zinc, that contact both of these substrates in the same manner, we then would expect an equivalent drop in both substrates, which would give us an FC1 effect of 1, so no differential effect. And when we actually did the experiment, we saw an effect only on the monoester reaction, which is consistent with the first model and telling us that Y112 is functionally coupled to R164. Now, we didn't just do this for Y112, we did it for all of the residues or all of the mutations we made, and we could ask how extensive are these functional effects connecting to these two active site residues. And so what we saw were these specificity effects kind of all over the entire enzyme. So just to orient you, K162 and R164 are here. Then Y112 is here, right next to R164, but we can see that around these residues, there's this density of functionally coupled residues with similar effects. And what's this telling us is that evolution has had to put in a lot of scaffold constraints around these two active site residues to get them to work to their full potential. And it's not just the second shell or nearby residues. These effects actually stretch all the way throughout the enzyme, even to the surface in some cases. So what we've done here is taken that big map of effects on kcat over km that I showed you in blue, and then we've dissected that into effects on two functional coupling to two different active site residues. We next ask the question, well, do different regions affect different catalytic mechanisms other than the specificity effect with R164 and K162? And I don't really have time to tell you the details in this talk, but by using phosphate affinity measurements, we were able to identify residues involved in a mechanism of catalysis called ground state destabilization. And this is shown here in blue. And the sort of most important take-home point here is that the regions of residues that I talked about with these FC1 effects is completely different than the region of residues that have the ground state destabilization effects. So the enzyme has these different regions associated with what you might call microscopic aspects of function. And it's telling us that evolution has put in a lot of work throughout the entire enzyme, and different regions are optimized to help do different tasks. So, and ultimately, what we were able to do in this and some follow-up work that came out last year was to go further and identify effects on additional and different aspects of function. So again, I don't have a great deal of time to go through the whole sort of project, but many of these mutations actually cause folding deficiencies. They cause the enzyme to irreversibly misfold. So we were able to separate misfolding effects from effects on catalysis. As I've told you about, we were then able to take those effects on catalysis and break them down into these specificity effects and these ground state destabilization effects. And then the follow-up work, we used transition state analogs to further probe features underlying specific recognition for the transition state over the other states of the enzyme. And so the main take-home points here are that it just highlights how intimately linked these distal residues are to catalysis and the various aspects of catalytic function. And it also highlights how complex function really is. So it's not just a matter of seeing a drop in catalysis. What we need is to understand the sort of physical basis in terms of the interactions between the scaffold and the active site that actually underlie catalysis. And so, as I said before, PAFA from a biological perspective is a little bit of a boring enzyme, and so from an industrial or biological perspective, there are lots of similar and perhaps more interesting questions to answer. And my hope is that HTMEC can be used to address those in a very interesting and novel manner. And I think there is huge potential for applying HTMEC to other enzymes. For example, similar quantitative studies of enzymes like polymerases, kinases, esterases, doing synthetic biology. So one of my interests is taking, libraries of natural sequences from fungi, bacteria, and seeing how sequence differences in those sequences ultimately translates to differences in function. And then, making enzyme design and enzyme optimization a little bit more rational. So instead of relying on, very laborious directed evolution experiments, can we sort of look at an enzyme structure and use these types of data sets to better predict sites we want to tune to get specific outcomes? And in terms of maybe you guys, looking at disease variants, at least in the coding sequences of enzymes, but of course, as we just heard, a lot of these mutations are outside of the coding regions of proteins and enzymes. So it can't really help much there. But for the ones that are, it's quite a useful tool. And then testing computational models, doing drug screenings, and lots of other fun things. And now, this is probably the most important slide, the acknowledgements. Obviously, a project like this, which coupled technology development with mechanistic characterization, was highly multidisciplinary and required an entire team, not just me, so a team of talented colleagues. Daniel McTarry, especially, who is a co-author of all aspects of this project. He's gone on to start a company, and is an amazing scientist and an amazing friend. Fanny Sunden's a really talented research associate in Dan Hirschlag's lab. We're building on a lot of her work, understanding how PAF-A functions, to actually be able to say anything about how these distal residues function. And then Mason Appel, Scott Longwell, they helped acquire data as well as made significant contributions to the experimental workflow and analysis. And then, of course, Dan and Polly, who were my postdoc supervisors and just amazing scientists and great mentors. And thank you all for listening. And I'm happy to take any questions.
Thank you, Craig. So thank you for the great talk. We have a question here. So what is your take on using machine learning tools like RF diffusion and AlphaFold to further weed out variants that are not predicted to fold properly?
It's a great question. So one thing, I actually still working with Polly and a student, Micah Olivis, in her lab, and we're actually looking at sequences designed by machine learning algorithms to ask if they predict function. I know that's a little bit different to the question you asked. So it's something I need to look into again. So when AlphaFold first came out, we took a lot of the mutations that affected both catalysis and misfolding. We ran them through AlphaFold, and we got the exact same wild type structure to like the sub-angstrom level. So either there's something we don't understand, or AlphaFold is just struggling to predict the effects of single mutations on natural enzymes. And I think there's some other evidence in the literature that that's the case. But I'll caveat that by that was AlphaFold 1, and I haven't redone that. But it would be really interesting to look at, especially the misfolding aspect.
Thank you. And another question from the audience is that since enzymes are trapped on the surface, how would one adapt HTMEC to enzymes that are dimeric? For example, MPRO from SARS-CoV-2 linker design, or is local concentration high enough that this doesn't matter?
That's a great question as well. I think I can't give you a yes or no answer. I think it's going to come down to sort of idiosyncratic effects of the system. Some anecdotal evidence that dimers work is that early in the project, we used E. coli alkaline phosphatase, which is an obligate tight dimer, obligate for function. And we were able to pull it down to the surface, and the rates we measured matched literature rates in solution. And so it suggests that at least for a certain number of enzymes, maybe particularly tight dimers, it doesn't seem to have any effects. That's one system. So this sort of answers like you have to try it. And every time we introduce a new system to HTMEC, we always try to pick, we do wild type, but also try to select known mutants from the literature and make sure we can recapitulate those measurements, expressing in the cell free, pulling down to the surface, having the EGFP tag, just to make sure that none of those ever have a problem, because, it's so idiosyncratic that you can't assume. And I think that's an important point.
Thank you so much. So I just want to thank Craig and Meek for the great presentation. And thank you everyone for attending the seminars virtually. And you can feel free to check out today's presentation on YouTube and previous talks on our YouTube channel. And I hope to see you guys back in October for our monthly seminar. Thank you so much.